hell is this thing called?
An animal.
Isn't there a saying that you could put lipstick on a pig and it's still a pig?
It's a pig.
You know, nope, that's a red-lipped batfish.
A red-lipped batfish.
So it's got red lips. That wasn't doctored.
Wait, those are the
Those are the lips.
Those are the lips.
The red part.
Oh my gosh.
And it walks on the ocean floor.
It's a, I need your DNA.
Oh wow.
Why Mother Nature?
So what's
Why?
the little thing up there? The little red.
Is it his nostrils? Like
Oh no, just
That's the nose.
no. That's a lure. It uses to feed with.
So this is.
It's just its front face. It doesn't have a nose. It has gills. It's a fish.
Yeah. Oh.
It's a fish. Scuba diving. Water.
Right, right.
Nemo.
Now I got it.
All right, come on. All right.
That's too good.
So, two important things you guys got to realize. Number one, males have a common pathway for both urination and reproduction. All right, females have two pathways, right? One for urination and one for reproduction. Males have one for both. Two, answer me this. Where do I go to look to find ovaries in a female? Where do I go to find them? I mean, where are they? Candace, what would you say?
Like in the pelvic region?
Okay, yeah. All right. Where are testes?
In the scrotum.
Down below.
Down below.
Is that in the pelvic cavity?
No. No. It's by the.
So you got to realize this. Yours are in, mine are out. Why is that?
Temperature.
Because which?
Temperature.
Really?
Yes.
All right. How much temperature, young lady?
How much temperature?
Uh-huh.
Regulations?
Yep.
I don't know the exact.
3 degrees.
Oh, 3 degrees.
Testes have to be about 3 degrees cooler than core temp. So evolution says I can fix that. I'll put them outside the body.
Wow. Sorry.
Okay.
That's 3 degrees Celsius or Fahrenheit?
Fahrenheit. Kelvin.
That'd be like Celsius.
Just drop out.
All right. So along with
Reagan is
that,
what you
then.
meant there. Yeah, Reagan.
All right. Oh, come on. What's wrong with you? Oh, it has to be plugged in. That kind of helps. Okay. Let's talk about those testes for a moment. The testes are actually formed and descend into the scrotum at 7 months of fetal development. So guys are born with them already in the scrotum. At least they're supposed to be. And when one or both don't descend, and they stay in the pelvic cavity, that's called cryptokidism. Crypto meaning hidden. Right? And you don't want that because you're in the body now. That means you're going to be 98.6 on average. That'll lead to infertility issues, poor sperm development, because they have to be cooler than core temp. So if a male is born and one or both testes haven't gone into the scrotum, the doctor says, well, let's give them about a year. Let's wait and see what happens. And usually they come down all by themselves.
That's 7th month of gestation.
Mm-hmm.
But it,
And
but it doesn't cause like long-lasting infertility or fertility issues if they're constantly spontaneously remaking sperm like every day.
well, they're going to be very poorly malformed and developed if the testes are still in the pelvic cavity.
But if once they drop.
Oh, no, once you're fine. Yeah, once you're up, they're fine. Yeah. And if they don't drop, then the surgeon can go in and simply just kind of push them through the inguinal ligament. Right? So we're kind of looking at, I need a different diagram to show you. We'll show you that one in a second. So 7 months of gestation, testes are formed. They don't come out, called cryptokidism. Now the covering of the testes has 2 layers of tissue covering them. And they're called the tunica vaginalis and tunica albuginea. But this is where things are a little bit different for you guys. You have to be careful. Think about serous membranes, right? When they line the inside, we give it a certain name, but then when the same tissue goes on top of an organ, we change the name of it, right? We're not doing that this time. Right? Here is the tunica vaginalis. It's this line right there. Goes up, comes around. I'd be tempted to change the name because now it's covering an organ, and we don't. Right? So lining the inside of the scrotal sac and actually covering the outer layer of the testes is the tunica vaginalis. If I peel off the tunica vaginalis, there's this white line right under it, right? That is the tunica albuginea because it means the layering, the covering of white. And that white line when it protrudes up into the testes, creates what's called septa, walls. And these walls help to create compartments known as lobules. Just like the liver, just like the pancreas has lobules. These guys have lobules. So, 2 layers of the tunica vaginalis. One is the outer one lining the inside, then there's the one actually covering the testes itself. Then under that layer is the tunica vaginea, which forms the septa. And we'll talk about the retate testes in a moment. Now this is a cross-section of testes. What they've done is they've taken the testes and cut them in half this way, cross-section, and we're looking up into the underside of our cross-section. That means I get to realize that my testes, it's not one scrotal sac for two testes, it's actually a scrotal sac divided into 2 chambers.
That's
So
a
each
coronal-
testis is in each one.
That's a coronal section, right? It's in a vertical plane.
It's a cross-section looking up into them.
Isn't it looking from interior because we see the epididymis?
Well, it's a bit of an angle
Okay.
to do it, because we're kind of coming down.
It doesn't matter.
Well, actually, no, you're right. It is a coronal. Yeah. But it can't be a coronal through one. That's the problem, because we're showing 2 separate testes. Now, these little purple ramen noodle looking like things, right, are apparently called semi-ferocious tubules, right? They're actually seminiferous tubules. Okay. I had a student call them semi-ferocious because he went semi-ferocious tubules. Okay. Right? But it's seminiferous tubules, right? It's in that structure we're going to make sperm. Now, remember how we gave you a representation of a uterus as a pear, right? For the right shape and the right size. A walnut is not a bad representation for a testes in terms of size. Now, I want you to imagine a walnut. Now, in that walnut, we have a bunch of seminiferous tubules. In each walnut, we have 2,100 feet worth of seminiferous tubules.
No.
How many feet to a mile?
5,280.
You have a half mile of tubes in each testes.
Oh my.
That's how tiny they are.
So there's a mile
That
of
is
you
insane.
combined.
I would think so. Well, not quite. You're short by about 1,000 feet, but you know.
Wow.
And you're saying in each
Testes.
lobe? Oh, it's in each testy. Okay.
Right.
Okay.
Each one of each. So there's about 200 seminiferous tubules per testes. Each one's about 80 centimeters in length.
Okay, but all together is that okay?
Yeah. So that's a lot of little semi-ferocious tubules inside of the testes. Now, this is a 3-dimensional view of them. And it's a little better than looking at a cross-section or a longitudinal section of them, because now I get to see kind of in some depth, but I still have the same layers. That light blue one is your tunica vaginalis. They've peeled it off so you can see under it. So the purple that you see is actually the tunica vulginea. Now I can see the walls that it creates for the septa. I get a better idea of compartments called lobules, and within them are the seminiferous tubules. And then with this picture, I can realize all of the seminiferous tubules will merge together into a structure called the retate testis. And then the retate testis will merge into fewer passageways called the efferent ductules. And the efferent ductules will lead to multiple tube-like structures forming the head of the epididymis. This isn't just one tube, there's many, many tubes up here. But it's going to form the region we're going to call the head of the epididymis for the moment. Then the epididymis will come down, and then it's going to form the ductus deferens. The other name for that is the vas deferens, hence for vasectomies. It's just going to run on the posterior aspect of the testes. It's going to go up, and it'll pass into the pelvic cavity, which we'll show in a couple of slides from now.
So is the spermatic cord completely enclosed in tunica vaginalis?
No, because
I mean, where does that stop? Because in the picture here, it looks like the tunica vaginalis goes up the spermatic cord.
yeah, it's sort of like the dura mater. It'll kind of just taper off and end.
Oh,
And we have a picture to show you that in a second anyhow.
okay. Okay.
So muscle tissue. Let's go back and look here. You've got this layer of muscle tissue right there. which is the dartos, and then you have this thicker muscle layer right here. All right? Now that's the cremaster muscle. The dartos is smooth, the cremaster is skeletal. It's the cremaster that plays a role in temperature regulation. So when testes get cold or the temperature drops even more, the muscle contracts, pulls the scrotum up to the pelvic floor to get them warmer. When they get too warm, the muscle relaxes, the testes descend, and get further from the body to try and keep their temperature at an average level. The spermatic cord is a wrapping of all of the tissue, all along through here, and we're going to have arteries, veins, lymphatic vessels, which we don't need to memorize the names of the arteries and the veins, but keep in mind the ductus deferens, which is the focus of this, is coming up through the spermatic cord along with all the arteries and the veins and lymphatic vessels. It'll pass through a canal, a little opening called the inguinal ring, and goes into the pelvic cavity, and that's the bladder. And we're going to go behind the bladder with this ductus deferens. Here they're showing you the cremaster muscle, so I can in my mind envision if the muscle is shortened, it pulls the scrotum up. If the muscle relaxes, it allows the scrotum to relax. So I can keep the testes at the proper temperature. They get cold, we pull them up. They get warm, they will descend.
No, you said that's a skeletal muscle, the cremaster,
Mm-hmm.
but it's not under voluntary control, so it's not that I can make it do it. So
I've never heard of a male who can voluntarily control their scrotum either.
Yeah,
You
so
tried
but-
earlier.
we tried.
You can keep trying to let us know.
All I can do
Just
is
we'll trust you. You don't need a video.
Yeah, all I can do is a Kegel exercise. Yeah, you know, I went through childbirth
No,
classes 3 times.
but that's the idea is that I want you guys to be thinking about that. Is that John says skeletal, I should be able to control that.
Is
And
that a reflex or?
it is more of a reflex. It's a quick reflex. I mean, cold air will make them draw up. Cold water, an ice cube. But
It's probably controlled by a reflex.
well, there has to be a reflex. I've never had anybody talk about the actual thermoregulation mechanism for it. But there has to be temperature sensors in the tissue, in the skin more than likely. And if the skin gets a certain temperature, that'll cause the muscle to contract. Definitely a reflex arc. In the testes themselves, you guys have two major groups of tissue. You're going to have the interstitial cells, otherwise known as the cells of Leydig, and they're going to produce testosterone, and we'll see the mechanism of that on Monday. And then we have our seminiferous tubules. Now, it's a tube, and the inside of the tube is lined with Sertoli cells, otherwise known as sustentacular. We're going to stop there for a second and come back to this. This is a diagram of a seminiferous tubule. There's the outer periphery of it. And then the purple stuff you see are the the Sertoli cells, otherwise known as sustentacular cells, otherwise known as nurse cells, because they're going to nurse the developing sperm. Now, this picture doesn't do it justice, because I don't really see individual cells. So I added this to your lecture notes. We can focus on this one, then go back and look at the previous one. That's the nucleus of one Sertoli. There's the nucleus of the other. And this is where you really want to look. Don't look, but look. And I realize that there is the cytoplasmic membrane of my Sertoli cell running all along here. It seems to be going around these little turquoise green things. What I want you to notice is all these little green things are not in the cell. The cell is like engulfing itself around them, like it's doing endocytosis, but they're not actually in it. And that's why they're called nurse cells, because they're nursing, they're protecting the developing sperm cells, which are represented by these little green circles. Now, the other thing besides protecting the sperm in sperm production, they form what's called the blood-testis barrier. It's a barrier between the blood and the testes, actually the developing sperm cells. That's because of your innate immune system. When do you guys create your innate immune system? When is it programmed? You guys remember? Well, it's formed while you're developing fetus. Your immune system is introduced to all different cells of you,
Oh, yeah.
right? What cell do I not have as a developing male? I don't have sperm cells,
No haploid cells, right?
right? So my immune system says, all right, I have no idea what the hell those little green rocket ships are. I've never seen them before. They must be foreign proteins. I'm going to attack and kill them with my immune army. So we need a barrier between the blood and these developing sperm cells. Because your innate immune system has never seen them before. That's the idea of a blood-testes barrier. Keep the immune system away from the developing testes. And to do that, we use these little yellow dots. Those are actually tight junctions. Remember, tight junctions are used to keep two neighboring cells held together. And we've always known that tight junctions, really for the most part, don't let things pass between them. Recently, like the last 5 to 10 years, we figured out that what happens is, and we'll walk through this more on Monday, my developing sperm cells, as they migrate from the peripheral edge where the stem cells are, they have to work their way down and get to the lumen. In order to do that, they're passing between two neighboring Sertoli cells. And to do that, the tight junctions have to dissolve. So the idea is that as a cell moves up to tight junctions, they break down, you move through them, then they reseal behind you. It's like walking through multiple rooms of quarantine. You walk into a room, you close the door, open up this door, walk through it, close this door, then open up this door. You just keep working your way down through the cells.
You need to show the Get Smart opening month. That'll make
No, that's true, okay? But that does form the blood-testes barrier. And again, because your immune system hasn't been programmed to recognize the proteins of sperm, and therefore they will attack them.
Does it stay that way forever?
Does what stay that way?
Where our blood want to attack our sperm?
Oh yeah, always.
Even right now?
Yeah.
At this age? Okay.
Yeah. It would create quite an immune response if I introduce your immune system to your sperm.
Just confirmation, so the blood-testing barriers is from the wall of the seminiferous tube.
So it's the tight junctions between the neighboring cells, because my blood supply is going to be out here.
Okay.
And remember, what can white blood cells do that red blood cells can't do? They can get through capillaries and walk through tissues, right? I don't want them walking into my seminiferous tubules and munching on my primary spermatocytes. All right, let's go back to that three-dimensional model of the testes. How many regions are there to the epididymis?
3.
You sure?
Yes.
Yeah, it says so in blue right there. All right. What are the 3 regions? You have the head, the body,
Tail.
and the tail. It's about 23 feet in length for all of that. Now it'll take about 2 weeks for sperm to travel down the epididymis, so they're going to be created, right, in the lumen of our seminiferous tubule. Then they're going to be migrating, right, through the retate testes, they swim up through the efferent ductules, into the head. It takes them about 2 weeks to work their way all the way down here and get into the ductus deferens. And there they are going to sit, like a baseball player on deck, waiting for their turn to leave. They'll spend many months there, if needed. And if not, if they're never used, they simply just die and get reabsorbed. We recycle all the parts so we can use those parts again to make whatever we need to make from those parts.
Takes them to go through there.
It takes about 2 weeks to get to the epididymis. Then they stay in the ductus deferens or vas deferens up to about a couple of months, 3 or 4 months. And by then they're going to just die. They've kind of gone through their expiration date.
Is that why the tube gets to the ductus deferens is so straight, just allows for like a quick getaway?
Yeah. I mean, it'd be really hard for us to try and, well, two reasons actually. I mean, that's a really good statement. Let's hold that thought and let's do this. Let's get to the ductus and the cross section, and we'll go back to your comment of why it's a nice straight tube. It's lined by ciliated epithelium. It's actually pseudo-stratified ciliated, not columnar like it is in the oviduct. Right? This is pseudo-stratified ciliated. And has a very, very thick muscular layer. Let's look at how thick that muscular layer is. So the next slide shows you, if you look at the actual histology slide, if I were a new student and I'm just doing what John says, look at a picture, I would say, oh, John, dude, that's simple columnar. Right? It's not. That is a poor cross-section that's supposed to be pseudostratified. I can tell, if I look right here, I can see the fuzziness of the cilia. And so I might be thinking columnar, but it's actually pseudostratified. So don't get fooled by the tissue appearance. But this is this, right? That's the lumen we have over here, right? And this entire gray area is smooth muscle tissue. It's about 6 times the diameter of the lumen. And that's because we have to propel from the ductus deferens. We have to push it up through the ductus deferens, behind the bladder, right, past the seminal glands, through the prostate, and out through the spongy urethra. Right? That takes a lot of muscular force to do this. So we need a really thick muscle to do so, and the ductus deferens has one of those. The other idea is the supporting glands to help nurture and provide a medium for the sperm cells. We have seminal vesicles or a seminal gland, it's the same thing. We have the prostate gland and then the bulbourethral, otherwise known as the Cowper's gland. In terms of volume, seminal vesicles provide 60% of the actual volume of ejaculate. And there's lots of fructose in there so we can keep the mitochondria going in the tail of the sperm. The prostate provides about 1/3 of the total volume. It also contributes a little bit of an acidic fluid and an antibiotic known as seminal plasmin. And that seminal plasmin helps to kill bacteria in the urethra, since it is a common pathway for urination and ejaculation. The Cowper's gland, or the bulbourethral, is about 5% of volume. Creates a and it's a really alkaline solution. I think it's around 4 or 3, I think.
That'd be acidic.
Yeah.
I'm sorry, you're right. That's 8 or 9 is what I'm trying to think of. My brain just went totally dyslexic there. That's because of the pH, the acidity of the vaginal canal. Now to see where they are, right? One section is this. Remember, this is the posterior backside of the bladder, right? Posterior side, up against the rectum, right? We're looking this way, and we can see the seminal vesicles or the seminal gland. So there's the ductus deferens. Like the fallopian tube has an ampulla, we have an ampulla for the ductus deferens, and then they will merge together and be joined by the 2 seminal vesicles to form the ejaculatory duct that then leads into the prostatic urethra, to the membranous urethra, and that's where we find our 2 pea-sized bulbourethral glands. So they will add their contribution into the beginning of the spongy urethra, even though they themselves sit next to the membranous part of the urethra. In terms of the structure of the penis itself, like a uterus that has parts, we're going to have 3 major parts. You have the root that connects the penis to the pelvic bones. You have the body, which will contain 2 layers of tissue, the corpora cavernosa and corpus spongiosum. Then the glands. The glands will be the head, and the body is the shaft. Now within that structure, let's go look at a cross-section of this. I heard you guys talking about it earlier. So there's the spongy urethra, right? And then there's the corpus spongiosum surrounding it. Then I have my 2 cavernosa columns that are highly vascular. When you look at it lengthwise, it'll look like this. There's the spongy urethra, with then the spongiosum on the side, and then the 2 columns of cavernosa with the crux, or the root, and there'll be actually ligaments going from here then to the pelvic bones, to the ischium and the pubis.
Slide 18 looked like that fish to me, kinda.
Slide 18?
Yeah, it kinda did.
What is slide 18?
No, totally.
Lost you a bit.
Yeah, slide 18.
Yeah, no wonder it's that one. Doesn't it kinda look,
Oh,
maybe you can see
yeah.
it.
Oh,
That's my own, no, that's my, oh no, Mr. Bill. My two eyes and my mouth, okay.
That doesn't look like a
Okay.
That's crazy. I got a question.
Yes.
What's the purpose of the prostate gland?
Mainly looking at the idea of providing that.
Okay. Oh yeah. But like, why does he get cancer so easily?
That's a good question. That I don't know. Other than the fact, my always quick response is, well, we've already lived past our useful normal lifespan anyways due to food and medicine.
Yeah.
I mean, most guys weren't having urination problems 20,000 years. They were dead by then anyways.
Yeah.
So we're probably just,
That makes sense.
or it could be the food. I don't know. I mean, it's a good question. I don't know why it's so sensitive to that. Is there another? Okay. So apparently then we could call this the
That fish.
red penis fish or something?
Yeah.
Okay.
Totally.
Yeah.
All right.
It's a Mr. Bill.
You guys are on your own. That was your last anatomy lecture. You can get your quiz scores from last Monday up here. You guys already know them for the most part anyways. There's an answer key on Canvas for you for this quiz since it was the biggest one. So you guys can see what the correct answers would be.